Today we're going to talk about a very important concept, and that is the structural composition of a protein. Now, I'm sure many of us have heard of an amino acid, right? All right. We've also probably obviously heard of what a protein is. Today we are going to connect the dots. We're going to connect what an amino acid is to a protein. Okay. Now, why is this important? What's the overall importance? Well, let's think of what proteins do. Proteins are going to be important for making things like antibodies. Okay, these are really important in our immune system. Proteins are important for making what we call a sarcomere. This we call a thin and thick filament model. All right, how our muscles contract is explained with proteins. How you are bringing in oxygen right now is through hemoglobin in your red blood cells. Okay, again, explained by protein. Enzymes in your body are digesting food and processing your food. Again, those proteins make up the enzymes. Many of your hormones, for instance, insulin, again, another protein. Okay? Point is, proteins are going to be very important. And understanding this relationship is critical in this chapter and the next chapter when we start getting into the genetic basis of how amino acids lead to protein. Really important for understanding a lot of genetic diseases. Now, I used hemoglobin down here for a reason, because this is going to be important for understanding things like sickle cell anemia, for instance. Okay, so we're laying the foundation for a lot of important material, both this chapter and the next chapter. So the first thing we're going to talk about is a primary sequence, and we denote it with a 1. Now, what is primary sequence? Well, think about a sentence. A sentence is a linear order of words, right? Okay, so there has to be, a sentence is like the cat and dog ate food. Okay, so each of these words is akin to a particular amino acid. And we have different words here, right? And the particular order of these words combines to make a sentence, does it not? It does. Well, the particular order or sequence of amino acids combines to make the primary sequence of a protein. For instance, the first amino acid might be one that we call methionine. The next one could be leucine. The next one could be isoleucine. The next one could be tryptophan. Okay? We could have many different types of amino acids. All right? Now, I've shown 6 amino acids here. In actuality, this can go on for hundreds and often thousands of amino acids. Okay, and the totality of this, the particular order of the amino acids, for example, methionine, leucine, isoleucine, tryptophan, methionine, you continue that linear order, that is the primary structure of an amino acid. It is akin to the order of words in a sentence. How we order sentences in a linear sequence is a sentence, right? How we put those words together. Well, how we put these particular amino acids together, okay, is the primary sequence to a protein. The next concept is the secondary sequence. So amino acids do not live in a vacuum, if you will, which is to say that this amino acid right here can interact with this amino acid right here. And similarly, this amino acid right here can interact with this amino acid over here. In a similar vein, this amino acid here can interact with this amino acid over here, and this one can interact with one we've not shown here. And these have a cross connection of interactions. Now, what's the consequence of this? Okay, here we show it like a chain, right? Well, let's just have this idea. What if these can connect? What can we make with this? What kind of structures can we make? This is what we call an alpha helix. So what's happening here? Well, this linear structure I've drawn, so it kind of looks like a curly fry, if you will, and each of these green nodules is an amino acid. Now, do we see how we have up here where this amino acid can interact with this amino acid? Well, let's just say this amino acid was to bond with this amino acid and form a hydrogen bond. And let's just say this amino acid could form a hydrogen bond with this amino acid over here. And this amino acid right here could form a hydrogen bond with this amino acid over here. And I'll just draw them in, but you're going to see that very soon that instead of having a linear chain-like structure, now we're getting some kind of three-dimensionality here. Now we're getting something that looks very similar to a spring, all right, or a curly fry. Okay, you sometimes see it represented like this. So think about how a spring would look, or a slinky maybe is a good analogy, or even I think a better analogy is a curly fry. You see, now that like has a three-dimensional shape to it. All right, we call that three-dimensional curly fry shape an alpha helix. Okay, this is one type of secondary structure. All right, here's another one. Now think of this as more as a flat plane-like structure. Now we're not talking about a curly fry type of thing. Now we're talking about a flat plane. And each of these nodules is an amino acid. And again, what if these amino acids are interacting with in a certain way that allows this more flat structure to form? Okay, we call this a beta sheet. All right, it forms a flat-like structure on the protein. This is another type of secondary structure. And then you might ask the question, why does a beta sheet form instead of an alpha helix? It all has to do with the type of amino acids. If you change a particular amino acid, this bonding characteristic changes and you form different types of structure. So it's really explained, or can be explained in a large degree, by the particular types of amino acids. Now, this is going to become important when we start talking about genetic defects. In that situation, that sometimes an amino acid may change, may become another amino acid. And when you have another amino acid, you break or you change this secondary structure, and that can change the entire function of a protein. Okay, so that's going to become really important when we start talking about things like sickle cell anemia. Sickle cell anemia, believe it or not, is from one defective or one different amino acid. And that one different amino acid changes the structure in such a way that that hemoglobin molecule behaves entirely different and leads to what we know as sickle cell anemia. So this is going to become important later on. Okay, but this is secondary structure. Now I have drawn the tertiary structure of a protein. The tertiary structure is the totality of the entire three-dimensional structure of a protein. Now, I've tried to highlight some secondary structure, for example, some beta areas and some alpha helix. Okay, but if you add all of those together, the totality, the entire three-dimensional structure of a protein is its tertiary structure. Okay, we're going to see multiple examples of this during the semester, but think of this as almost a big clump of clay, for instance, right? You know how a clump of clay has three-dimensionality to it? Okay, and all the nooks and crannies and cavities in that big clump of clay. Okay, the protein is the same thing. The protein has all sorts of cavities and nooks and crannies, and we'll talk about active sites later on. But that protein has three-dimensional shape. Okay, that three-dimensional shape is by a large part the combined interaction of all of these secondary structures. Okay, all those secondary structures combine to make the tertiary structure to a protein. Okay, now this is important when we start talking about how a protein functions, because if you change the tertiary structure of a protein, you're going to change the function of a protein, or at least you can change the function of a protein. Doesn't mean you will, but if you do change the tertiary structure, you very well may change the function of a protein. The fourth level of structure we're going to talk about is the quaternary structure. Now, this structure is not present in all proteins, but only some. And in this situation, we're going to have two or more proteins connect to make a functional protein. The quintessential example of this, and we'll talk about this, of course, is hemoglobin. Okay, we have a heme group here. Hemoglobin. This hemoglobin is in your red blood cells, and this holds oxygen. And this is going to carry oxygen through your body to your tissues. All right, this is a four-part protein. So we have four what we call polypeptide chains, and these all connect, and as they connect, they make one single hemoglobin. All right, this idea of having two or more polypeptide chains to make a functional protein is the quaternary structure of a protein. Now, not all proteins will have this. Okay, only a few proteins will have this structural model where two or more polypeptide chains have to combine to make a functional protein. So let's review what we covered. We said that the primary sequence was the linear order of amino acids, right? Okay, like words in a sentence. That's our primary sequence. Each dot here being a different amino acid. We then said that these different amino acids can interrelate with each other, right? They can interconnect. They can bond with each other, and that formed these alpha helices, right? And it also formed these beta sheets. Okay? So what I'm trying to show is this linear line up here looped around itself to form this, and folded up on itself to form this. Okay, this is the secondary structure. Now, this secondary structure will combine to make a larger protein. All right. When it combines to make a larger protein, that is the tertiary structure. So the entire three-dimensional shape of that protein, which is made up of things like alpha helices here, made up of those beta sheets here, that is the tertiary structure to a protein. Finally, in some cases, we're going to have two or more proteins combine, or in this case, you probably more refer to them as a polypeptide chain. And in this case, you will have them connect to form a functional protein. I'm trying to draw hemoglobin here. This is the quaternary structure of a protein. And again, not all proteins will end with this. Many proteins will actually end with the tertiary structure here. Only a few proteins will have this quaternary structure to it. All right, so those are the levels of protein organization.